Good morning, everyone, or good evening. This is the first lecture in male reproductive system, the histology of the male reproductive system. These slides were made by Dr. Hadwa, but I'm Dr. Shayma. Today we will discuss the components of the male reproductive system, the different components. Then we will describe the structure of the testis. Then we will describe the cell division. This is a very important topic, the cell division in the testis, meiosis and mitosis. Then we will describe the structure, the change in the structure during spermiogenesis. Meaning of spermiogenesis: sperm, which is the sperm, the male sperm. Genesis is the formation. So spermiogenesis is the formation of the male sperm. Then we will correlate structure to function of the blood-testis barrier, which is a testis between the blood vessels and the testis. Then we will describe and correlate the function of the Sertoli cell. Sertoli cell is a supporting cell present in the testis. Then we will describe the structure and function of Leydig cells. Leydig cells are some cells present in the connective tissue of the testis. The structure of the male reproductive system, you have to learn it very well. It is formed of testis. This is the testis. The testis is connected to the epididymis. The epididymis is the site for sperm storage. Then it passes to the vas deferens or ductus deferens. Then the vas deferens would go to form an ampulla and then enter into the prostatic urethra. In the prostatic urethra, this is the prostatic urethra. This is the urinary bladder, and this is the prostatic urethra. And there is also the seminal vesicle opens at the end of the ampulla. And then you have the bulbourethral gland, and it opens into the penile, the beginning of the penile urethra. This is the part of the membranous urethra. This is the penile urethra. Okay. In the penis, this is the penis. The penis is formed of corpus cavernosa and corpus spongiosa. The corpus cavernosa are the ones that are present outside. The corpus spongiosa is the one that surrounds the penile urethra. Okay, then again you have the testis. This is the testis. Then you have the connection. This is the mediastinum testis or the reti testis or whatsoever. We will discuss it later in the next lecture. Then you have the epididymis, which is the reservoir. You have the duct deferens or vas deferens, and the vas deferens and the seminal vesicle open into the prostatic urethra, whereas the bulbourethral gland open into the penile urethra. Again, this is the testis. Testis is covered by skin. This is the skin. Okay. And this is the tunica vaginalis. It contains minimal fluid content. Tunica vaginalis contains minimal fluid content. And you have the testis itself. The white color surrounding the testis is the tunica albuginea. The tunica albuginea, which is a tough connective tissue layer. okay? And then you have the things that contain blood vessels. It is called tunica vasculosa. And you have here, this is the epididymis, this is the epididymis, this is the vas, the beginning of the vas deferens. This is something called the spermatic cord. As you can see, the spermatic cord is formed of artery, lots of veins in the vas deferens, plus muscles. Okay, we'll discuss it later. Then the testis is covered by tunica albuginea, tunica vaginalis, then tunica albuginea and tunica vasculosa, and this is the epididymis, which is a reservoir for the testis. Again, this is the tunica vaginalis. It contains minimal fluid. It is like a balloon. It contains minimal amount of fluid. This is the tunica vaginalis. Okay. Then you have tunica albuginea, which is a whitish, dense connective tissue layer. Then you have underneath it tunica vasculosa, which is loose areolar connective tissue rich in blood capillaries. There are lots of septa in the testis that separate the testis to form the lobules. Lots of septa to form the lobules. Each lobule contains a seminephrous tubule, which is present in lobules. Okay, and of course you have connective tissue, reticular connective tissue in between. This is the structure inside the testis. You have tunica albuginea, tunica vasculosa, and seminephrous tubule in the testis lobules. Okay, the seminephrous tubule are present about 2 to 3 seminephrous tubule in each testis lobule, testicular lobule. The seminephrous tubule is formed of lots of cells lying on a basement membrane. They are the spermatogenic cells. They all lie on a basement membrane. Okay. Since the seminephrous tubule form a tube, so if you have a cut section of this tube, it would appear as a circle. It would appear as a circle, like in this slide, this part, as you can see here. This is a seminephrous tubule. You have, this is the basement membrane, and these are the cells, the spermatogenic cells. Okay, the spermatogenic cell appear to be 4 to 8 layers of cells. They are cells in the testis during different differentiation phases. Here again, this is the seminephrous tubule. As you can see, this is the basement membrane. Okay, this is the basement membrane. If you form a circle here, then it would be the seminephrous tubule itself. Okay, this is the connective tissue in between the seminephrous tubule. As you can see, you have here myoid cell, something called myoid cell. We will discuss it later. And this is the connective tissue cell, another Leydig cell present in the connective tissue. Okay, anyway, you have here about 4 to 8 cells present. These are spermatogenic cells in different differentiation phases. And you have a large cell called Sertoli cell. Okay. Each seminephrous tubule is highly convoluted. It is about 30 to 70 centimeter in length. 30 to 70 centimeter in length and 0.2 millimeter in diameter. Okay, it have a well-defined basement membrane and is surrounded by connective tissue from the outside. The connective tissue in the outside contain The connective tissue in the outside contains the myoid cell that give rhythmic contractions so that it would help the spermatids and the sperms, the newly formed sperms in the seminephrous tubule, to gain their function. The lining of the seminephrous tubule, there are two lineages, the spermatogenic lineage and Sertoli cell lineage. We will discuss now the spermatogenic lineage. Okay. The spermatogenic lineage, you have 4 types of cells. You have 4 types of cells. Here on the base you have spermatogonium, either type A or type B. Type A you have dark and pale, and type B, the pale spermatogonia, pale type A, would give rise to type B. And all of the spermatogonia divide by mitosis. The DNA content inside the spermatogonia is 46 chromosomes. Okay. From the beginning of the primary spermatocyte, the B spermatogonia will give rise to primary spermatocyte. The primary spermatocyte is present in the compartment called adluminal compartment. You can see this is a process coming from the Sertoli cell. Divide the primary spermatocyte from the spermatogonia. Again, you can see there is a process here between the spermatogonia and primary spermatocyte. Again, here there is a process. The Sertoli cell divides the seminephrous tubule into 2 parts. Part at the base, it is called the basal compartment, and part at the lumen, it is called the luminal compartment. The primary spermatocyte is formed of 46 chromosomes, but it would enter meiosis. So it would enter the S phase, and then it would give rise to secondary spermatocyte, which is 23 D chromosome, and go further into meiosis 2, and form early spermatids, which is 23 S chromosome. The early spermatid would change their shape to form the late spermatid or the sperms. Okay. Then again, you have here, this is the basement membrane of the seminephrous tubule. You have in the luminal part, you have the spermatogonia. You have two main types of spermatogonia, type A and type B. Type A is divided into dark and pale cells. The pale cell divide into to give type B spermatogonia. The type B spermatogonia give rise to primary spermatocyte, which is 46 S chromosomes. Then it enters the meiosis S phase and form 46 D chromosome. The secondary spermatocyte would form 23 D chromosomes and would enter meiosis 2 to form 46 S chromosomes. And then the spermatids, early spermatid, would change their shape to form the late spermatids or the spermatocytes, the sperms themselves. Okay, okay, through differentiation. This is a spermatogenesis, the spermatogonia, A or B. Okay. spermatogonia. It is formed of 2N, it is 46 S chromosome. Then it would form the spermatocyte, 2N again, which is 46 S, but as it enters the meiosis, it would change from 46S to 46D. This is meiosis 1. Then the cell transfer into meiosis 2. Meiosis 2 is the secondary spermatocyte. At the end of meiosis 2, it would form the spermatid. Then the spermatid change into the sperm, mature sperm, via differentiation phase. So there are 3 phases that take place in about 64 days. In 64 days, the spermatocytogenesis. The spermatocytogenesis is the formation of the spermatocytes. This is the spermatocytogenesis. Then you have spermiogenesis, meiosis phase, which is meiosis 1 and meiosis 2. And then you have the spermiogenesis, which is the process of differentiation and transformation of spermatid into sperms, into mature sperms. This is the formation, the spermatocytogenesis, spermatocyte, the formation of spermatocytes. Genesis, which is formation, means formation. So spermatocytogenesis is the formation of spermatocyte, and it happens via mitosis. So 2N cells form another 2N cells. There is no difference here. Whereas in the meiosis, the spermatocyte change into secondary spermatocyte. This is meiosis 1, and then it would change into spermatid, and this is meiosis 2. After meiosis, the spermatid change its shape from the rounded structure to form the mature sperm. This is the spermiogenesis. This is the spermiogenesis. Here again, the spermatogonia change into primary spermatocyte. What was the name of this stage? This stage name was spermatocytogenesis. Spermatocytogenesis. Then the primary spermatocyte would change into secondary spermatocyte and change into spermatids, early spermatid. This is the meiosis, and then the spermatid would mature into sperm, mature sperm. This is spermiogenesis. Okay, so from this stage to this stage, it is called spermatocytogenesis, meiosis, and then spermiogenesis. This is the spermatogonia. The spermatogonia are present in the basal compartment of the seminephrous tubule at the base. The spermatogonia, you have pale type of spermatogonia and dark type of spermatogonia. The dark spermatogonia are reservoir. They are reservoir, okay. Whereas the pale type spermatocyte, they are the types that would enter mitosis. They would change into type B spermatocyte, and they would form themselves again. So they change into 2 types of cells, either themselves or into type B spermatocytes. The type B spermatocytes, this is the cells that would enter into the luminal compartment to change into spermatocyte and to enter mitosis. They renew themselves and multiply by mitosis as well. So we have 3 types of spermatogonia. 2 of them are called type A and 1 is called type B. Type A you have pale spermatogonia and dark spermatogonia. The pale spermatogonia undergo mitosis to form type B spermatogonia, which enter mitosis again to increase in number to form the primary spermatocyte. These cells appear oval in shape with spherical nuclei, and they have densely stained chromatin. However, during mitosis, the chromatin, especially in the here, you have darkly stained chromatin in the dark spermatocyte, whereas in the type A pale, the chromatin is less dark or less electron dense because it will enter mitosis. Okay, and you have lots of ribosomes present in these cells. All of them are 46 S chromosomes. All of them are 46 S chromosomes. Type B spermatocytes divide by mitosis under the effect of FSH hormone to give rise to primary spermatocyte, and this happens during puberty. It happens at puberty. For the primary spermatocyte, it originates from B spermatogonia, okay, and it is present in the beginning of the luminal compartment. It is the largest cell in the seminephrous tubule. Okay, this is the primary spermatocyte. Most of the cells that you can see in this photo are primary spermatocyte. They stay for about 22 days, so they stay for a long time. It's the content of the DNA present in it is 2N at the beginning, but most of the primary spermatocyte have 4N because they have entered S phase, so they have 4N, which is 46 D chromosomes. Okay. They would enter the prophase of the first meiotic division, which would take about 22 days. The prophase of the first meiotic division is formed of 5 stages. These 5 stages are called, number 1 is called leptotene. The second stage is called zygotene. Then you have pachytene. The fourth stage is called diplotene. And the last is the diakinesis. Okay. Here again, as you can see, this is the seminephrous tubule. This is the basement membrane. These are the spermatogonia. This is the Sertoli cell. You have Sertoli cell processes separating the spermatogonia from the primary spermatocyte. The primary spermatocyte contain 4N. Most of them contain 4N because they enter the meiosis stage, so they pass through the S phase. Then the primary spermatocyte, after 22 days, more than 22 days, they would form the secondary spermatocyte, which would finish the secondary meiotic division without any S phase before it. So it is short-lived and quickly move into forming of the spermatids. And all of these cells are embedded inside the Sertoli cell. The primary spermatocyte and second spermatocyte are embedded in the Sertoli cell. You have the Sertoli cell form two compartments, basal compartment and luminal compartment. Here in this photo, this is the Sertoli cell, and in the basal compartment there is in the basal compartment there is spermatogonia, whereas you have junctional complexes here, and the primary spermatocyte is formed in the luminal compartment. For the Sertoli cell to form this junction, it creates a junction above the cell, above the dividing cell, and another below the dividing cell, and it would release the junction above and complete the formation of the junction below, so that the cells can move freely from the basal compartment to the adluminal compartment. The secondary spermatocytes, they originate from the primary spermatocyte. They are small and oval. The DNA content in it is 2N, but they are 23 D chromosome, because they will go through the second meiotic division. They would enter the second meiotic division to form the spermatids. Okay, for the spermatids, they are 1N. Their DNA, the DNA content inside it is 1N, 1D. Okay. They have lots of ribosomes. They have well-developed Golgi and numerous mitochondria. No more cell division would happen in the spermatid. No more meiosis or mitosis. This is the end stage for the formation of sperms, and then there would happen differentiation into mature sperm. Okay, here you can see this is type A dark. Type A dark by mitosis give rise to type A pale. Type A pale by another set of mitosis form type B cells. Type B cells by mitosis again give rise to primary spermatocyte. The primary spermatocyte by meiosis, meiotic division, they would give rise to secondary spermatocyte and then early spermatids. The DNA content in all of these cells is 2N, which is 46 S chromosome, whereas from the beginning of the primary spermatocyte, the cell becomes 4N, which is 46 D chromosomes, and in the secondary spermatocyte it would be 2N, which is 23 D chromosome, and in the early spermatid it would be 1N, which is 23 S chromosome. Okay. As you can see in this photo, there are some bridges between the cell, between different cells. There are bridges, like here. The cell don't lose their connections. There are always bridges between spermatogenic cells, okay? And they don't lose their connections except after the sperm loses its connection with the Sertoli cell, and the bridges remain forming the residual bodies that would be phagocytosed by Sertoli cells. So again, the dark cell divide by mitosis, giving pale cells, pale type A cells. Then pale type A cell divide again by mitosis, giving rise to type B cells. Type B cells, by the effect of FSH during puberty, it would form the primary spermatocytes. It would undergo meiosis, and it would form the spermatids, and the spermatids via spermiogenesis would form mature sperm. So For the spermatogenesis, the formation of the sperm, okay, for the spermiogenesis, the change from this phase to this phase is formed via something called spermiogenesis, formation of sperm, okay. This stage is formed by different consecutive stages of proliferation and cytological changes from early male germ cells to mature spermatozoa. Okay, they are either spermatocytogenesis, meiosis, and then spermiogenesis. Spermatocytogenesis was the formation of sperm, primary spermatocyte. Meiosis is the change from the primary spermatocyte into spermatid, and then the spermatid into mature sperm. It is called spermiogenesis. Spermiogenesis is divided into 4 stages. These 4 stages are Golgi phase, cap phase, and acrosome phase. Then at last the maturation phase. The main structure that is present here is the Golgi. The Golgi form specific protein, and this protein is stored in the Golgi apparatus, so it is called a cap. Then the cap would extend itself to form the acrosome. The acrosome is the structure present at the head of the sperm, and it contains hydrolytic enzymes to enter into the oocyte, into the zona pellucida of the oocyte. And then the maturation phase is the phase where any residual bodies, any parts, any excessive parts in the sperm would be removed to form the final shape of the sperm. So you have 4 stages: Golgi phase, cap phase, acrosomal phase, and maturation phase. This is the Golgi phase. In the Golgi phase, a small PAS positive granules appear in the Golgi. Okay, in the Golgi. Okay, so these granules accumulate more to form the cap. Then the cap would change and form a larger structure which would be called the acrosome. Okay. The DNA content in the nucleus would condense each time more and more, condense more, and the centrioles here, the 2 centrioles one of them would go to the lower part of the sperm, and the other would go upward and it would disintegrate. The lower centriole would form the basal body of the flagella, and it would form the axoneme here, which is 9 multiplied by 3, 2 central microtubules. Okay, and all of the mitochondria would surround this axoneme. All of the mitochondria would go to surround the axoneme, forming the middle piece of the sperm, and the excess cytoplasm present here would form the residual body. The residual body would be phagocytosed by Sertoli cell. Okay, then it would form the mature sperm. The structure of the mature sperm The structure of the mature sperm is formed of a head which is 5 micrometer in diameter, middle piece 5 micrometer in diameter, principal piece which is the flagella 50 micrometer in diameter, and then the end piece which is again 5 micrometer in diameter. Okay, this is the Golgi phase. The Golgi phase, you have small PAS positive granules. that are present in the Golgi. They fuse together to form the acrosomal vesicle. The two centrioles, each one of them move in different direction. Then in the cap phase, the acrosomal vesicle flattens and expands to form a cap on the anterior half of the nucleus. This is the cap on the anterior half of the nucleus, and it is called the acrosomal cap. Okay, it contains hydrolytic enzymes so that it can pass through the zona pellucida of the oocyte in order to pass all the way into the interior of the oocyte. Only the head of the sperm can enter into the oocyte. The acrosomal phase, there are lots of changes happening in the immature sperm in the spermatid. There are lots of things happening in the spermatid. Okay. First of all, the nucleus elongates. And the anterior pole would be capped by acrosomal cap. The centriole forms the neck for the developing axoneme of the flagella, which would be the axoneme. This axoneme would be 9 doublets and 2 central singlets. Okay, the second centriole mostly would fragment. There would be 9 coarse fibers that are formed and extend around the microtubules. These are the fibers. Okay, these are the dense fibers that surround the axoneme. This is the axoneme. The axoneme is formed of 9 doublets, 2 central singlets, and there are coarse fibers, and then you have mitochondria present on the outside. Okay, and they stop, the mitochondria surrounds them, and it stops at something called the annulus, which is the end of the middle piece. At the end of the middle piece, you have here something called the annulus, and this is where the mitochondria stops. Any excess cytoplasm would be present in the posterior region, and it would be called the residual body, and it would be phagocytosed by Sertoli cells. For the maturation phase, the Sertoli cell phagocytose the residual body, and the mature sperm become free from the Sertoli cell into the lumen of the seminephrous tubule. So once the sperm is free in the lumen of the seminephrous tubule, then the maturation phase has started. For the part of the sperm, for different parts of the sperm, there is the head. This is the head of the sperm, and then you have the middle piece, you have the principal piece of the flagella, and you have the end piece. The head is about 5 micrometer, middle piece 5 micrometer, 50 micrometer, and then 5 micrometers end piece. The head contains the nucleus and the acrosome. Okay, it appears pyriform in shape, and once the nucleus touches the ova, it would a reaction called acrosomal reaction would happen, so that the nucleus become activated, and the DNA inside it would be activated to fuse with the ova. And then you have the middle piece. The middle piece contains the axoneme of the flagella, and it have also 9 longitudinal fibers and mitochondrial sheath, which we said that the mitochondrial sheath ends at something called the annulus, and of course it is covered by cell membrane. Then you have the tail, or the flagella itself, the tail, which is 50 micrometer in diameter. The principal piece is formed of axoneme and fibrous sheath, which are circumferential, whereas the end piece is only formed of axoneme only axoneme. So the tail is formed of axoneme and fibers, circumferential fibers, whereas the end piece is formed only by the axoneme itself. What are the factors affecting the spermatogenesis? The first factor is the temperature. It needs lower temperature by 3 to 4 degrees Celsius than the body. So the testis is present outside the body in the scrotum. And then you have the effect of different hormones from the anterior pituitary, FSH hormone or the LH hormone, or it is called the ICSH, which would influence the interstitial cells, okay, which is the Leydig cells. The testosterone secreted by the Leydig cells would affect the spermatogenesis. Any dietary deficiency and systemic diseases or toxins would also affect the spermatogenesis. In a case of undescended testis, this is an undescended testis. Testis stay in the inguinal canal or even higher in the abdomen. It needs early intervention during the early months of post-neonatal life. So the surgeon would take this testis to put it in the scrotum, in its place, in its normal place, so that spermatogenesis would not be affected. This condition is common in premature males. It can lead to irreversible histological changes in the testis and increase in rate of testicular cancer if it is kept inside the abdomen, and it requires urgent surgical correction by placing of the testis into the scrotum again. Again, we have said that the seminephrous tubule is formed of spermatogenic lineage, which we discussed, and then we have the Sertoli cell. Okay, for the Sertoli cell is a main supporting cell present in the seminephrous tubule. It forms the Sertoli to Sertoli junctions, and it also forms Sertoli to spermatid junction, okay, which is released when the sperms are released into the lumen of the seminephrous tubule. This is the Sertoli cell. The nucleus of the Sertoli cell is pale, which indicates an active cell. Okay, it is a very active cell, Sertoli cells. They are very large. They lie on the basement membrane, and they form the luminal compartment and the basal compartment. By electron microscope, the Sertoli cell contains rough endoplasmic reticulum. It contains also smooth endoplasmic reticulum. It contains Golgi apparatus. It contains lysosomes. It contains microtubule for support. It contains everything that could be present in any cell, in any active cell. It has junctions. The first junction is Sertoli to Sertoli junction that forms the blood-testis barrier. And it also has Sertoli to spermatid junction that by its release it would allow the maturation of the or donate the maturation of the sperm. Here again, this is the Sertoli cell. The cell that is present in the basal compartment. What is the name of this cell? This is the spermatogonia. either A or B spermatogonium. Okay, then you have the first cell after the blood testis barrier. This cell is the primary spermatocyte. Then you have secondary spermatocytes, spermatids or immature sperms, and then here late spermatids. And if there is one here, this is the sperms. What is the function of Sertoli cell? It would form the testicular fluid, and it would have androgen binding protein to bind to testosterone to deliver this testosterone into the testicular fluid. It have inhibin hormone, which has negative effect on FSH. It have Mullerian inhibiting hormone, which act its function intrauterine life to inhibit the Mullerian tubes to change this baby into a male. It have testicular transferrin. Its main function is for support, protect, and nourishment for the different spermatogenic cells. It have a phagocytic activity to phagocytose different residual body, the phagocytose residual body. It help in the formation of blood testis barrier via tight junctions, and there are also gap junctions between the cells and each other. Sertoli cells do not divide during their reproductive life, and they are resistant to X-ray infection, radiation infection, and malnutrition. They are resistant cells. Here again, this is the blood testis barrier. The blood testis barrier forms the basal compartment and luminal compartment. The basal compartment contains the spermatogonia, whereas the luminal compartment contains spermatocytes. This is the blood testis barrier. Okay, it is formed by Sertoli to Sertoli junctions. What is the function of blood testis barrier? Its main function is to protect the spermatocytes from the immunity of the body. The spermatogonic cells, their structure, their DNA structures is 46 chromosome like any body cell, whereas from the primary spermatocyte to the spermatids there are crossing over and variation in the DNA content. The DNA content in them is different from the DNA content of the host or from the male. So if antibodies would see this DNA, it would suggest that this DNA is not from the body, and it would attack it. And therefore, if the blood testis barrier is severed and it is defective, the antibody would be formed and pass to the spermatocytes and cause infertility to this male. So, and if the sperm content would pass to the immunity, into the body immunity, to the plasma cells and other immune cells, it would form sperm-specific antigen, and it would create immune response that would be very hard to treat and to protect the body from it. So the spermatozoa and the spermatogenic cell possess molecules that are unique to these cells and are recognized as foreign by the immune system, by the male immune system. The spermatozoa are first produced at puberty, long after the individual have become immune competent. So failure of spermatozoa to remain isolated in during the production of sperm, they would cause immune reaction, severe immune reaction, and it would form something like vasectomy and a special cause of infertility in males. That's it. I hope that you have understood the difficult topic of male reproductive system, and in the next lecture we will continue the tubes of the male reproductive system. Thank you.